All right, gang, it's 2:02, so we're going to get started. And we only have an hour and a half for this session, so we really got to get started. Kind of joking, but yeah. All right. Okay. I think I asked originally for 40 minutes, and they gave me an hour and a half, so, but this seems like a chatty group, I can tell, so this will be good. Yes. So my name is Mason. Okay. Um, cool. All right. Thanks for coming. I'm Mason Heberling from Carnegie Museum. We have Will Weaver here who is going to kind of lurk behind me awkwardly, we decided, and he'll chime in too. We'll see how we can do this. But this is a discussion session. I don't think I participated in a discussion session at one of these, and now I'm hosting one, so we'll see how it goes. But I'm really excited to do this with Will, and I think Rob Gorelnik might hopefully join us in the virtual space, and maybe we'll have his head up there with sage wisdom at some point. But yeah, so I'm excited to talk a bit here about trait data from herbarium specimens. My background is actually, now that I'm a museum curator, I of course love herbarium specimens, but my background is as a functional trait ecologist, and that's really what brought me into the herbarium space. So as many of you may know and do know, herbaria are all about, and biological collections at large, but we're focusing in on the plants, herbaria. Archive diversity across scales, so taxonomically, spatially, temporally, both across species and within species. So here's a map showing more than 3,500 herbaria distributed across the world of more than 396 million specimens and counting. And so we oftentimes think of herbaria as these kind of centers for taxonomic research, and they very much are. But they also have the potential, or already are, and have the potential to become more of repositories for trait data, and in particular thinking about functional trait ecology. And just to give a little bit of a shameful plug for my own work that kind of like inspired me wanting to do this session, because I have never actually presented on any of this work before, was a couple of years ago I wrote the Coulter Review for the International Journal of Plant Sciences on the very topic of herbaria and plant research. And it was really fun, so check out this paper. I spent like definitely multiple years accruing all the different studies that I could get my hands on, so not really all that systematically, acquiring all the different studies that use herbarium specimens to measure functional traits. And I thought it would be cool to have a session, to have a lot of different perspectives, a lot of different people here to talk about trait work in herbarium specimens, what's been done, what's the current state, what's the future, and then in particular, what are the limitations and what can we do moving forward with existing specimens we have, and then also new collections. So the digital extended specimen, wow, I'm doing really good on time here. It's 2:05, so I hope you guys are really had a nice lunch and are ready to talk. So the digital extended specimen is really a lot about traits. So on the right, the Webster kind of the genesis of this extended specimen idea, with the bird specimen surrounded by a constellation of different data streams, either relating directly to that given bird specimen or on a more larger holistic scale to relating to the species or other co-occurrence records, et cetera. And so this has been definitely developed across many different biodiversity collections. And this past year in the Herbarium Handbook, with my colleague Bonnie Isaac, we wrote a real little like, ooh, does that mean Rob's here? No. Okay. It's like a doorbell. We got to write a little bit about extended specimen. I was really inspired by this bird figure, and we made one for herbarium specimens. And I'm sure many of you can think of the many other different kind of connections with with the extended specimen concept, and even just a single given herbarium sheet, whether that be connections to iNaturalist, digital record, historical climate data, species associations, phylogenomic data, insect collections that may relate to a given plant, for instance, historical records, and then also putting it all together in kind of data aggregators like GBIF. But what I didn't say was the number 4, the tri-trait database, and thinking about extracting traits from a given specimen. And what I'd like to have this discussion session be here, oh my goodness, I go in and out, okay, is really thinking about, as we start to extract all these other forms of information from the specimen, what can be extracted and what do we do with it? So a couple of years ago also, the rise of functional traits in herbaria. Here's just a figure in one botanical journal, the American Journal of Botany, in the last just couple of decades, looking at the number of papers. Here, looking at the use of DNA from herbarium specimens and also functional traits. And you can see that really in the last 20 years, there's been kind of this explosion of this relatively new use, or at least repackaged use, of herbarium specimens for functional traits. Sorry, I'll get comfortable. And we're about hour 20 minutes into it. I'll be really comfortable with this. That'll be good. Okay. So can herbaria fill gaps in trait data? Here is one big global trait database called TRI. I think I'm told, I don't know if anybody here might be able to confirm this story, but the genesis of the TRI is just like, we'll try to have a database that accumulates all the different trait data. I really think that's the story. So that's kind of interesting. So they tried and they've largely succeeded. And so as of online, you can go on the tri-trait database. There's many different data sets on there. We're pushing 15.5 million trait records across 305 plant taxa, of which many of these traits, many of you may be familiar with, kind of these big leaf economic traits in particular. How are you doing over there, Will? You're good? Okay. So if we were to just kind of expand from TRI, so thinking about here showing the total number of records in the TRI plant trait database, and then let's just assume that maybe we're able to extract one trait from an herbarium specimen. And I think maybe some specimens we might not be able to extract any traits, but some we might be able to extract more than one. So just humor me here in this comparison. So let's see, as of 2 years ago, the number of herbarium specimens with images on GBIF was, pushing 35 million specimens with images. Here also showing those that are georeferenced and non-georeferenced, just as a reference point. Cue for laughter. And you can see that GBIF has a much, just in total number of records, many, many more than the tri-trait database. So we have an opportunity here. And then looking at the number of species, here breaking down TRI into kind of three kind of big traits, specific leaf area, leaf nitrogen on a mass basis, and leaf area. The number of species that have at least one record in each of those 3 traits. And let's make the very rough assumption that maybe on average we might be able to get these 3 traits from a given herbarium specimen, and with there being over 400,000 at least taxa, let's say, not necessarily species, represented in GBIF as of a couple of years ago. And then thinking intra-specifically within plant species, here showing the number of observations per species, so this kind of like within species replication. In TRI, the median is about 2. Whereas we know that herbarium specimens, we oftentimes, even within a given collection, we have many, many representatives, right, of the same species, perhaps even collected in the same location or nearby. So the take home here is there's a big opportunity here, right, to fill the gaps in the tri-trait database. And Rob had this great kind of breakdown thinking about the different sources of traits from specimens. We can measure traits from the digital images. We can measure traits from the digital images. We can measure traits from the physical specimens, or we could do a combination thereof. And then you can also get traits from the label text. Sometimes the label has some interesting. And of course, here it depends what you define as a given trait, but for instance, reproductive status or even other other kind of, especially kind of discrete or categorical traits like flower color. And so here is just one example paper that's pretty cool that was published a couple of years ago, The Effects of Climate Change on Floral Anthocyan and Polymorphisms. And the entire paper used this label text for flower color. So whatever the collector wrote on the label, doing a really cool analysis of floral color polymorphism based from the herbarium record, but from those kind of other notes, if you will, on the herbarium label. All right, so what can we get from specimens? And I want to open this up. We'll open it up soon. Hopefully I'm not boring everybody too much. It's a hot crowd, we're good. Thinking about what traits can we get from specimens. And so like I said, when I wrote this review, I really got really obsessive and was trying to include all of the different things I could possibly think of. And then even since then, I see new papers coming out all the time doing new measurements on herbarium specimens, which is really cool. So here I'm just going to kind of go through kind of some big categories, but I want to open it up and see if people have other either known examples or new aha moments of traits that could be extracted from herbarium specimens for biological research. So first, whole plant traits like plant height, growth form, spinescence, just as kind of three examples. Here's on the right side, a cool paper from over 20 years ago now that used herbarium specimens to look at American ginseng, and there's kind of a little drawing recreation of the results of looking at decline in whole plant size with over-harvesting. And then I had to throw this paper in too because it's a really cool one too that by Buswell et al. In Australia in the Journal of Ecology. And this paper is actually what kind of got me into herbarium work, believe it or not. A really cool paper looking at herbarium introduced plants, looking at the herbarium record, asking the question of is rapid evolution common in introduced plant species, where they look at many traits, including whole plant height traits of herbarium specimens of species that fit on the sheet, of course, and finding differences over time as a given species spreads from its initial points of introduction. All right, and then leaf level traits like leaf size, shape, and SLA. I think, Will, you have some thoughts on leaf size and shape that you'll share here later. You don't need to right now. Okay, cool. Vein density, vein patterns, leaf chemistry, nutrients, heavy metals, isotopes. There's like a handful of papers for all of these different traits, which is really, really cool. Reflectance is another big one. I think I even put, so there's been a lot of research coming out in the last couple of years using, looking at leaf spectral reflectance and looking at how that correlates to other given traits like SLA or leaf nitrogen, for instance, doing these non-destructive methods of surveying many species in herbaria for various traits. And I like that picture because it's kind of cool. It's like, you just got a little trait gun and you just push the button and you get answers. I think it might be a little bit more complicated than that, but I like the idea. And then stomata and trichomes also, which is some work that I've been involved in more recently, looking at the changes in poison ivy in particular, which is kind of a mild obsession of mine as of the last couple of years, looking at the impacts of increased carbon dioxide on poison ivy. All right. And then, of course, phenology. Sometimes we always separate phenology from functional traits, but they do fit in there. And there's like this kind of a whole body of work with phenology and herbarium specimens that this slide certainly, although it's a great paper, where's Kate? Yeah, it's a great paper, but there's so much great work happening with phenology and herbarium specimens. And I think even that hasn't fully been realized, but has certainly been realized more than a lot of the other traits. All right, and Will, I mean, do you want to talk a little bit, Will, or you want me to just say, we didn't really plan this too well. Yeah, you'll talk about it. Okay, Will's going to talk a little.
Okay, so one of the projects I've been working on quite a bit is Leaf Machine 2, and the goal is to extract as much quantitative trait data as we can from primarily leaves, but we also identify other organs in the plant, like roots, flowers, buds, um, basically anything we could find on an herbarium sheet. We also identify archival components, so we can find weights, labels, rulers, barcodes, a bunch of different archival components. So there's a bunch of different things you can do once you're able to extract that kind of information. We all might care quite a bit about the actual quantitative traits, but you could also do some interesting historical research once you are able to extract just label data, for example. If you have a really big collection, you could find every record from a specific collector, for example, even if it's hard to do that manually. Yeah, so this figure is basically just from our publication, and it just shows that with modern machine learning techniques, you can extract just about anything you want if you have enough training data to do that. So we've spent, I don't even know how many years getting all of this trait data together. So if you're working on just a genus or an individual species, it might not be worth it. You have to figure that out. The amount of annotations you need to create to do something specific might actually be bigger than your test case, for example. So we have to think about things that we're not normally used to thinking about for all of this work. So I guess I would just highlight that in an herbarium specimen, there's a lot of information, and some of it might seem unnecessary sometimes, but you can do some really cool things with each component. So yeah, we can talk more about that later.
Thank you, Will. We'll get to the question portion here. So yeah, cool. So I guess a big question, and again, we'll certainly want to discuss it more, but thinking like, not only what could we theoretically extract from herbarium specimens, but is the data, are the data that we're extracting reliable and comparable to other trait protocols? And a lot of these trait protocols, believe it or not, the ideal isn't measuring a specimen or a leaf that was collected over 100 years ago, but instead is on fresh material. So there really are a lot of kind of legitimate concerns about measuring traits from herbarium specimens without knowing exactly how necessarily that specimen was collected and if that's comparable. And so a couple of years ago, with at the time graduate student at University of Miami, Tim Pires, asked this very question, basically making new collections and measuring traits from the same plants using fresh material, and then also from the dried material prepared like an herbarium specimen. Looking at, on the top, specific leaf area, the ratio of from area to mass. So that top figure there showing measuring fresh whole leaf area over dried mass, which is the standard for measuring specific leaf area, versus, actually it's not shown here, we also did a comparison of using hole punches, but here this is showing using a whole leaf that's already dried and doing that comparison, and the dashed line is the 1-to-1. So you can see there's a little bit of digression from the 1-to-1 line, but it's pretty good, all things considered. Same with leaf thickness, and then also branch-specific gravity at the bottom. And so each of those given points was a different species. And so here's just the leaf thickness. Each point is a species. This is the figure that was in the middle in the last slide. So we have this pretty tight 1-to-1 relationship for leaf thickness across species. But then with herbaria is really cool because we have a lot of replication, right? So here's within species doing this same comparison, so the same species. You can see it's a pretty good line, R squared 0.95, 95%, but a bit of a displacement from the 1-to-1 line, which is something to consider, but still exciting. And so, yeah, and I'm sure it's all of this, a lot of this has already come up in some of the other talks that I've seen today. There's a lot of emerging tools in herbarium research, and in particular herbarium trait research, some of which, Will had already talked about, and then also some other work by Ryan Falk and others on other methods. Here's one cool paper of a way of extracting kind of trait information from floristic data or from floristic treatments, which can also be done from herbarium specimens label data, like I mentioned before. All right, so specimen uses are changing, kind of like really getting into this functional trait work, and the question is, should we? So now that we have these kind of unanticipated specimen uses, how we engage with existing specimens might be rethought, and then also rethinking how we collect now. And so one thing I've realized coming into the herbarium space is that nature is more than fits on an herbarium sheet, certainly for some species more than others. And then Will, you added these nice little frowny, I don't know if you wanted to comment on your frowny cartoons you added. But I think just the idea that our biodiversity documentation is of course incomplete and imperfect. Not to say that it needs to be all thrown away, but it's something that needs to be considered. How's that for your? Okay, cool. And one method that we've been doing at the Carnegie Museum where I work is kind of connecting iNaturalists with herbarium specimens. And this is tangentially related to traits, but it certainly relates. Thinking of, certain aspects of a plant isn't preserved well in 2D and isn't preserved well dried or isn't preserved well pressed to a sheet a decade later. And so, botanists have long been taking pictures and photographs of the living organism or the living plant that they have actually, also collected. But oftentimes that's not necessarily connected with the given specimen. So we're kind of left, 100 years later, not quite sure if that particular plant was multi-stemmed, for instance, or if the color of the pollen was red or not, et cetera. And so iNaturalist provides this kind of really cool opportunity to not only tap into the iNaturalist community, but also in the functions therein, but to also kind of have a relatively efficient way of connecting biodiversity observations associated with that specimen images from the field. So yeah, that's what I said. Pictures capture aspects specimens can't. And some other things to consider too before we jump into the discussion. I can tell like half the room is really on the edge of their seat. We're getting there. And that is herbaria don't capture all life history stages well. And this is something else that been really actively thinking about we oftentimes collect plants when they're in flower or fruit and kind of an ideal specimen maybe isn't necessarily collected vegetatively, whether that be because it's difficult to identify or confirm its identity. But I think, that is part of the biodiversity spectrum that we want to be documenting. And one cool example are these life history vouchers. I'm going to go through a couple of kind of deep tracks of herbarium methods that are not well practiced but have been previously developed. And one of them is this concept of the life history voucher, where you might collect plants at different life stages. Snapshot vouchers, I'm definitely going to have Will talk about this because it's some cool stuff.
Yeah, so this is sort of an idea that tries to address a problem that Mason just mentioned, which is a lot of times, an herbarium might be hesitant to accept sterile specimen vouchers, for example. So if you're going to the field and you want to collect a specimen, there could be more diversity than you logistically have the capacity to take home with you, for example. So one thing we could potentially do is just start prioritizing digital-only collections a bit more. So not as a replacement, but sort of augmenting what we end up having on hand. So if you went out into the field, you might take home 10 vouchers, but you might image 200 at the same time to try to capture more of the breadth of what you're seeing in the field. So one thing you could do is something like this. We created some photogrammetric templates. They can be scaled up to any size you want. So we have some that you could use to image like a Kentucky coffee tree leaf or a palm frond. You could scale that up, image them in the field, and then still take home your voucher, but we all know that a palm frond is going to get folded up or cut up or something to mount. So this is a good in-between, and because of all of the advances in computer vision and machine learning, you can still end up doing really cool automated approaches with something like this. So part of it is being able to introduce some of the standardization that official archival vouchers have, but being able to take that into the field with you a little bit more.
Cool. Yeah, and then here's more, right? Cool. And more.
Oh.
Yeah. Oh, yeah, this seems like something you want to talk through.
So one other thing you can do is you could use this to create time series examples. So this is just something I did in my backyard to make sure it would work. But basically with with Field Prism, you can automatically correct distortion of the images. So if you're just in the field, you can take a photo with your cell phone. It doesn't matter what orientation you're in. So that's kind of going back to here. Every image starts out on the left, but when it goes through the pipeline, it ends up being, rectilinear to the camera, so you can get measurements from the images, even if they started out skewed to begin with. So doing that, you could end up doing some sort of time series approach without a lot of effort, basically.
Yeah, and I could see some curators maybe not wanting you to collect each of those. Cool. And another old approach as I was writing this paper that I legitimately just stumbled upon, and maybe some of you already know, this idea of like mass collections, or what others have called local population samples. And as a functional trait ecologist, kind of trying to think of ways to archive trait variation. This is from a 1941 publication by Edgar Anderson here, basically advocating a method of kind of systematically collecting in a given population, doing a transect and systematically collecting trait variation, whether that be leaves or some other aspect, and then basically throwing it all in a Palm folder, which is kind of an interesting idea. And some of these collections still exist, although not very many, having called some herbaria and trying to track down some of these mass collections. And I think some of them don't exist because they're kind of like curatorially difficult to maintain, or like, why did this person collect all these? You just make one standard voucher. So it didn't necessarily really take off, but I think now we might want to revisit some of these cool ideas. And here's one. This is a relatively less uncommon example of this mass collection, and this is all on a standard sheet, but each leaf is from a different form of this thimbleberry, I believe, rubus. And on the label, which is blown up there, you see the number of individuals within a population of the different forms. So not necessarily, I don't want you to get hung up on this particular example, but just the general idea of archiving all of the diversity, not necessarily just the mean, but all of the trait diversity. All right, I love this quote, and since I have an hour and a half and I have your uncaptivated attention, I had to read this. This is from Woodson, 1947. Okay, you ready? A false impression widely current among non-taxonomists is to the effect that herbarium specimens usually are collected because of some abnormality which attracts the fancy of the collector. The accusation reveals such prejudice that one is baffled for an effective retort. Perhaps a denial that falls far short of revealing the sincerity of a plant collector, but may impress the critical outsider, is the fact that plant collections habitually are made in multiple sets bearing identical serial numbers for the purposes of sale or exchange amongst numerous botanical institutions of the world. It would be difficult indeed for even an unusually perverse individual to pursue his passion under such adverse circumstances. Don't you love this? I love it. The chief danger in plant collecting actually is that of choosing too many, quote, normal specimens. The statistical errors from such likelihood, however, should be ineffective. Anyway, thanks for letting me read that, because I thought it was it's a really interesting point. And it's something that always comes up in the trait work with herbarium specimens, is this general idea of oh, but that was just collected because it's weird, or oh, that was just collected for, everybody kind of has these preconceived notions, and some of them may or may not be true, but I think now's the time where we need to kind of really rigorously assess that, rather than just being you can't measure herbivory from herbarium specimens because collectors are avoiding those specimens. And it's do we know that? Is it true? Anyway, so yeah, 1947, this was a nice quote. And in that same paper, I also this is Deep Tracks, and maybe some of you know this paper, but it's really interesting looking at measuring leaf shape from herbarium specimens. But I love this phenocontour map, and I've never actually really seen it anywhere else. But it's kind of cool looking at within species variation across the species range. And again, this is 1947, and I think with new tools and tools, methods, approaches, perspectives, there's just so much potential that many are doing, but there's just so much more potential that herbarium specimens have, and that's the in part motivation for this session. Edgar Anderson also went on then, this is your history, an abbreviated history of obscure herbarium trait archival techniques, is this idea of what he later then called inclusive herbaria. And you'll see from this specimen, this maize specimen from Missouri Botanical Garden, there's actually not any physical plant material on the specimen, which is kind of interesting. But in his book, Plants, Man, and Life, he talks about this inclusive herbaria method and published a few other things on this general approach. But it really does get back to this idea of really quantifying trait variation within a given population and species, which is kind of interesting. So Anderson had kind of this setup when he would go to different, I think in Mexico, do these kind of rigorous trait protocols of to look at the trait variation within different variations of maize in this case. And that's something that I've been kind of thinking, and I'm not saying I definitely found the solution, but I definitely get a lot of eyebrows raised from others, like, what are you doing? Why are you doing this? These aren't specimens. But it's been good fun. And then this idea that kind of combining, trying out some of these old mass collection methods with the life history methods, also trying to capture phenology, all of the same population. So this requires many different visits to the same population. And I'm not saying I necessarily would recommend this all to be like this, but I think in many cases, and there are many specific cases where at least doing this for at some level is really important and could be useful. All right, I think this might be my last quote, and thank you for humoring me again on my historical quotes, largely from the same sources. But I love this one too, also from Edgar Anderson from this 1952 book. Making a good herbarium record is something like trying to stable a camel in a dog kennel. So with that, I think we're getting to our discussion. I love that idea, and it's basically trying to think of the idealized specimen, even just assuming you have all the resources in the world and you could make, and even in the extended specimen concept, what would the ideal look like if you had all the time and all the money in the world? So with that, let's get going on a discussion. What do you think, Will? Okay. Did Rob make it or no? Rob made it. Hey, Rob.
Yeah, Rob is on as a panelist, and also just to let you know, you have 28 participants online.
All right, awesome.
So myself and Eleanor will keep track of the Q&A and let you know if there's any comments coming through from those folks. But
cool. So I have a couple of slides here with kind of questions, guiding questions, and if memory serves, we've kind of divvied them up into some different categories, but it's an open room. How much time we got to? Where are we? Oh my word. Okay, we're good. Okay, I can keep going. I can pull out some more obscure quotes if you want. All right, so traits from herbarium specimens. I kind of wanted to talk with the group, thinking about, strengths, weaknesses, missed opportunities. So I'll just read these questions and then we can just kind of riff from there. What traits are most amenable to herbarium specimen measurement? Which traits or species can't be measured and why? Is there anything we can do about that? And are there any missed opportunities? Are there traits that we're overlooking? So yeah, okay. Anybody have anything they want to say? Do it. All right. It's kind of hard to do a discussion session in this kind of very formal, but let's do it.
Yeah, thanks so much. I'm really curious about the ability of the algorithms that you're using to pull out data about fungal pathogen growth on leaves, the presence or absence of pathogens there that might be of interest and searchable in a database. And then secondly, the extension of this to other two-dimensional specimens outside of herbaria, like blood smears, for example.
Sure. I think as far as machine learning algorithms go, it's a matter of training data still. Um, you can teach machine learning algorithms to find outliers to some extent, but for something like fungus growing on a leaf, I don't really know. There have been a couple studies that have looked at that, also herbivory and things like that. But it is kind of a matter of what kind of training data you have, how consistent is the target species or the target data set to that type of approach. Because it could be the fact, like if you only have 1,000 images to analyze, it might take that entire data set to train something to be able to do the process you just did manually. So that's something you have to kind of think about. But I think if it's in an image, it's fair game, so to speak. Sometimes you have to be creative. Like if you're training a classifier to predict the class of an object or something like that, sometimes you have to think like a computer and not like a botanist, for example, and realize that it's just seeing texture. So something that might be an apparent difference to us might not be as obvious. You might have to combine classes or things like that. I don't know, does that kind of help? Or oh, you could do both. And if your goal is just to find those instances, that's definitely doable. If it's determining something more specific, that takes a little bit more. And I found that it's also easier usually to break it into pieces. So finding out, it's easier to train a classifier, for example, to say, is this infected or is it not? And then once you have all of the infected, then you teach it to discriminate amongst those and then add some in back from the beginning. But it's usually easier to break things into components and try to simplify as many decisions as it has to make. And there's also techniques you can do to sort of purposefully stratify your data. You don't always have to use the color images that we're looking at. You could do augmentations that make it more apparent for a machine learning algorithm to do something.
I might have missed this earlier because I came a little late, sorry. But is there, so you're talking about all these ideas of training, training machine, for machine learning and all that kind of stuff and the intense effort that goes into it. Is there like a hub or like a clearinghouse for these types of things to not duplicate effort? Like, the idea of training an algorithm to see a healthy plant, I mean, that should just be where everybody starts, and then they can train the algorithm to do something else, I don't.
Not yet. I think we all want to get to that point. There are a lot of open source machine learning data sets on the internet, and I think all of us would love to have that. Part of the problem is, like, for Leaf Machine's training data, I don't own the herbarium images, so I can't really just re-upload that somewhere. I don't feel comfortable doing that. So then you have the issue of you have to have links to things. Links break all the time, so even if you make it public, is someone going to be able to get all of the images that you used anyway? So we have a little bit of an issue just because if you're making a training data set that will work across institutions, you need multiple institutions' data sets to be able to do that. So that goes into a discussion we could probably all have, which is how do we deal with that? Because there's not a real easy solution for that. I guess sharing links is the best way, assuming the links are always going to work, which we know they don't. So yeah.
So for the folks online, Randy, I'm sorry, this is Deb Paul. Randy was saying maybe we could include those in our collection management system, the links. I was going to suggest that we talk about this at Tadwig, spinach, that's coming up in Okinawa. It's a very Tadwig type of question, biodiversity information standards. How are we going to share what we're learning so we don't reinvent the wheel, Randy? Yeah.
And that is true. I believe in the component-based analysis a lot. Like Leaf Machine is 7 or 8 different machine learning algorithms that you all join together. And some of those data sets are a lot simpler to create. So like just putting bounding boxes around things, that's pretty straightforward. And it's easy to get, a method that a lot of people could replicate. Things like segmenting leaves or segmenting flowers, that's a much more hands-on approach, and it takes a lot longer. So something like that, it would be great if we could start incorporating things elsewhere. So we need to develop standards. We have to figure out how to share all of those data and things like that.
So my name is Doug Boyer, and I work with Morphosource, and we've recently done something kind of like this with Harvard at the MCZ. We're hosting, I guess, what you in a way you might call sort of a deep integration or deep aggregation with their slide histology scans. So we use essentially we do sort of a two-legged query to import and migrate records through GBIF and through a IIIF server that they have. So the end user functionality of this is you can go in and use all the search and filter and query tools to essentially pull histology scans, histology images from Morphosource, but they're actually coming from Harvard's MCZ. And the way that the way everything's set up, I think it actually would work really well with herbarium sheets as well. So it's a protocol that we have developed that's pretty. There aren't a huge number of requirements for in terms of server setup and stuff that the institution would have to do that kind of integration. And then you'd have what you'd wanted. You could search the same taxon and pull in all the records from 5 different universities or something.
Yeah, that's great. I'm glad that we're getting to that point. So then it's just more about buy-in at that point, right? Because the problem that all of us deal with is the fragmented ecosystem for all of these projects. We all keep making our own little databases, data sets, and then trying to integrate them all back together again. So
yeah, that's right. And I think this builds on community standards already. So the guts of this is Darwin Core, GBIF, audiovisual core. So basically using those systems in a best practice manner will be, and as well as IIIF, which is like a more and more broadly adapted open image server framework, will make this. That's what's needed to use the system that we've set up for sort of aggregation and access.
I think Deb has something.
Just really quick, I wanted to add the question to you and to Rob online. What standards are being developed, biodiversity information standards, not just for the AI part, that also the standardization of the way we categorize this data, the trait data itself?
Yeah, I think that's a question Rob is best suited to answer.
Rob, you should be able to unmute yourself and talk.
Yeah. Guys, can you hear me okay? I'm a little worried. It sounds like a little bit of buzz.
Oh, yes, we can hear
One
you.
second, Rob.
It's a little too loud in here.
You're very loud.
Okay, I can come is it better from back here? How about if I whisper? Now I'm afraid to talk.
There we go. That's
we're
better,
good now.
Okay. Hello, people I cannot see. Deb, that's a great question on trait standardization. This is such an interesting one because it kind of subtends across a bunch of communities, and every community has their own trait definition. So if you're talking to like an insect community or a mammal community or the botany community, there are very different complexities around like how to define trait and trait measurements. And a lot of this comes down to remembering that we have to do these trait definitions, but we also have to talk about observations and measurements that allow us to link the traits we're describing, the entities and qualities, to observation processes, right? My quick take on this is like at one level, observation and measurements, like that's not as hard a problem to solve, maybe, and that there are lots of good examples of observation and measurement ontologies that are already out there. At the community level, like kind of coalescing traits, that's a bigger kind of pull together floristic traits, but there definitely are different ways that people conceive of what they're considering traits. I think of phenology as a trait. Others may think of phenology more in the context of phases or other ways that people kind of like build their conceptions. But I think at the heart of this, like this is a standardization process that every community needs to go through. Like we've done this for plant phenology via the plant phenology ontology. The big value to me isn't so much the trait standardization itself. Maybe it's what you get from knowledge systems. And so one reason why I've been really passionate about the idea of building ontologies in particular, like control vocabularies that have really strong hierarchical representation, is a lot of what we want to do is data integration, which really revolves around being able to kind of like take data that are coming at different granularities and working to assemble them in a way where we can discover the content we need across those different kind of granularities of how people report traits, whether people are reporting like the number of or percentage of leaves that are in a certain phenological state, or whether reporting presence or absence. That kind of aggregation process is really enabled by having these ontologies for traits. And so, I mean, there's no simple solution here. It really requires communities to kind of assemble to standardize. And I don't think, I mean, I don't know for sure, guys. I can't remember. I don't think there are like a single source plant trait ontology out there. I know there's the plant anatomy ontology, but I don't think the traits themselves are necessarily compiled into like one format, one ontology. So that's still work to be done, I think. It's weird being online, but all you hear is the silence of people being like, either what, or maybe like, oh, cool.
Sorry. Great. No. Yeah.
Hi. Hello. So I just had a quick question about picking your training data. So from my understanding with machine learning, it's identifying correlations, and so you might actually be identifying a correlation that you don't mean to. So for example, berries, they're squishy, no one wants to mount them, so then it's going to think every single plant that has berries is only flowering because that's the herbarium specimens that they have. So how are you guys dealing with that when you're picking your training data?
That's a great question. This actually just came up on a project I'm helping on. So like in this case, it was a classification project. So we just need it to tell us which of like 6 different classes a specimen is. If you do something like that, and you're working with a relatively small set of data, but if it was from 3 different herbaria, there's a chance that one class only exists at one herbarium. So then it sees that herbarium label, and then it classifies it as something else. So there's, that's a pretty obvious one, but in that case, I think I talked about it in a different one, but we can censor those labels and you just hide it to keep it from doing that. There's internal things you can do in a machine learning network to sort of remove some biases. So usually we just say, get as many samples as you can and label as many as you have the resources to label. Generally, you can get something working with a few hundred, maybe 1,000. You can just see if it will work. But there's mechanisms you can do for something like classification. If you have a long-tail distribution, you can change weights in the model so it downvotes the really common things. There's also ways to augment your data set. So you could just sort of create artificial data sets. So something that we've been thinking about is like there's certain ruler types that are pretty rare. But once we see them, you can just extract those and insert them into other herbarium images. So you can do image augmentation ahead of time to sort of fill in any gaps. For berries, you theoretically could do that. You could segment out berries and then just put them wherever you want artificially. You kind of have to use your imagination sometimes, but usually the problem is class bias in data sets. And because we're dealing with what we have, it's often difficult to fix that. So you kind of have to deal with it from an algorithmic side a lot of the times.
Sorry. I've got another question too. How have you guys ever played around with doing this, not just for vascular plants, but like for lichens and bryophytes and some of the smaller ones?
I guess at that point, it's which traits are you looking for? If it's just like area, that question would be relatively straightforward, potentially. But there are certain traits that just don't really make sense so to use an herbarium specimen for. So like one would be like stipules or trichomes. Sometimes those are just too small to see with a standard herbarium image. So I don't know, but if it's mining label data, then yeah, there's absolutely a lot you could do with that. So even compound leaves come up a lot, where because the leaves are usually too big to even fit on the sheet, most of the time you can't really even get enough training data to deal with compound leaves because they don't even fit on the sheet. So then you're dealing with leaflets and just treating them like a full leaf. So a lot of it is data set specific. Does that kind of help? Like what would you be looking for in one of those data sets?
We're trying to get microscope images for this purpose because I know that when you just look at the herbarium specimen, it's dried and it's crushed and they're tiny. They're so small, right, especially when you're taking a picture in a light box with standard herbarium specimens. But I'm just wondering, like, if we do put in the effort to get all of this library of compound microscope or dissecting microscope images, would that be something that machine learning could actually help with?
Yes. So in that case, you would just want to keep your standardization approaches in mind. So for Leaf Machine, I wanted it to be useful for any given herbarium collection. Because of that, we had to show it all of those different stages. So if you're dealing with microscopic images, if you want that to translate to external data sets, you would want to make sure that when you're imaging them, maybe you take it at different magnifications, for example, just so you get them all covered. So that's one thing to think about. You can do that manually by taking different resolution photos, but you could also potentially do that just by cropping in and out sometimes. So that would be one of the augmentation strategies. So it kind of just depends. If you're just working on one species from one collection, you don't have to think about that much. But if you want it to be more generalizable, you kind of have to think about all the variation you could see. And maybe someone didn't use exactly the same protocol. The lighting has shadows that yours didn't, something like that. So some of those you can compensate for with after-the-fact augmentation. Sometimes you want to do that ahead of time, too.
Perfect, thank you.
So I have a question that I think both of you guys may have some comments on, and maybe Rob will as well. So one of the things that herbarium specimens are super valuable for is inferring ploidy, either from pollen or from stomatal size. But it's really hard to get that sort of information out of our standard images. So I guess I was wondering, since you've both done work on leaves from specimens, and I think, Mason, you mentioned stomata as well, what sorts of imaging approaches might be necessary to be able to measure the stomata? Because we've worked with a computer scientist a bit to try to use different lightings and imaging approaches so that we could actually visualize the stomata in a way that we could try to infer ploidy, but it wasn't wholly successful. So I'm just wondering if you guys have some suggestions for that. Thanks.
Yeah, that's a great question. Yeah, I mean, my background with the measuring stomata from herbarium specimens is just taking casts from the leaves, like with nail polish. So looking at it under the scope. In terms of like actually like from digitized images, I have no experience with that. I don't know. Yeah, I don't know. Maybe someone else in the room has some experience or insights or thoughts on that. But yeah, that would be cool. Yeah, so that's not a very informed answer other than look at it under the microscope individually, each one. But yeah, maybe someone else has thoughts.
While I'm walking over to Deb, if anybody online has questions or comments, please put them in the Q&A.
So somebody was talking about dyeing things with iodine earlier, and I'm not sure this would work for old specimens, but would there be a way to see stomatal size? Can you run dye in a plant and see the stomate with a dye? You don't think so? Wait, yeah, Pam. Yeah, okay. What's a thought?
Yeah.
I know.
See, that's what I want. I want this outside of the box thinking. So yeah, I don't know though. Yeah.
Well, I was trying to imagine, and I know next to nothing about it, so you have to ask other people. I know you talked about heavy metals, things like the ability to see gold. I mean, if you look at a specimen with a certain kind of imaging, I don't know how that works, you can tell that there are metals present.
Right.
So I thought, well, do the metals hang out in the stomates?
Yeah.
If the metals hang out in the stomates, you could see the metals with the image, and then you could infer the size of the stomate because you can see the metal. So that's kind of where my brain was going with the dyes.
Yeah, that's a cool. Yeah, I have no.
But I don't know where the metals hang out.
No expert opinion on that other than cool. We've got online there. Anything? No, nothing. Okay. All right. So, Will, do you want to what? Oh, there is one. Oh my goodness.
Oh, good. Oh, good.
Oh,
good.
We're so excited. Okay.
From Nikki Nicholson at Q, you mentioned specimen duplicates in your intro talk. Is there scope for using specimen images from separate herbaria which come from the same collecting event to verify automated trait extraction?
You have thoughts on that?
I haven't done anything with that yet. That is a really fun idea. Yeah, duplicates are really interesting. I think not a lot has been done with them, at least from what I can tell, to link them all back together. But
yeah, I think with traits though, that it's kind of this middle area now of, is it actually a duplicate? Meaning, is it like the exact same individual, or is it another individual from the same population that may have a very different trait expression? So it's kind of like, I don't know. And these are some of the questions of like, well, if you have and maybe Will, maybe you have thoughts on it. It's like, okay, when it actually comes, I mean, all these ideas sound really great and fun, but then when you get an herbarium specimen, which most are just this huge jumble of leaves, of overlapping leaves, you want to measure leaf area, and you have all kinds of like, leaves at the tip versus in the middle versus, so like, what do? Do you measure all of them and take the average? Do you just measure the middle one? And how do you record that, I guess, what you do? And I'm kind of digressing here a little bit because I feel like duplicates are a similar case like that. It's like, well, do you consider it how do you, I guess, in your actual statistical model, how do you, you probably should account for the fact that it's collected in the same location at the same time, or same location, same species type of thing. But I don't think you could actually really use it as maybe in the way that maybe you could use it for other information from duplicates as like kind of like, just because traits are variable. But I don't know. Yeah.
Mason, Rob has his hand raised.
Oh, Rob. Yes, Rob.
Oh, no, I lowered it, but I think it's a fascinating idea, Nikki, and I really like the thinking behind it. I think what's really neat there is sort of separating out all the issues that Mason just raised, I think, rightly, that there are going to be challenges, but also like taphonomically, those will have been maintained separately for long periods of time by different institutions and collections, and that itself could actually create variance or variety that you want to like account for. So I think it's like really cool because you can start pulling apart like what is the biological signal there, what is the signal of just like literally taphonomy of being maintained in different collections that might itself create variability that you're not expecting. But I think it's a great idea. Really.
Yeah, I love that, Rob. Now that I think about it too, yeah, we were doing a project looking at, microbes associated with roots, and there seems to have been some, like a herbarium signal, like of different microbes in different herbaria, which is kind of interesting. I didn't even think of that. So yeah, that's it. I'm intrigued though about the duplicates thing and where to go from there with that.
Are you familiar, Mason, with the clustering algorithms that GBIF has employed to find the duplicate herbarium records?
I'm familiar with it in the sense of that you've just mentioned it. I've heard of that.
So Nikki was part of writing the algorithms that do that.
Yes, I knew that. I knew, Nikki, you were into duplicates. I knew that.
Yeah.
Yeah.
So.
guess that's sort of also related to taxonomic uncertainty and like finding specimens that have been misidentified too. That's a related issue that definitely needs to be addressed. One of the most popular things people have done with herbarium specimens is trying to do automated identification. And I think a lot of the approaches have been pretty successful, but one of the biggest problems is still rare taxa. And that's a doubly hard problem because there's very few people in the world that are even qualified to tell the difference anyway sometimes. So again, machine learning is basically pattern matching to an extent. So if you have something that's super rare, it's even more difficult to try to figure out what it is. So that's an interesting thing. But yeah, the duplicate issue is a really interesting sort of like pure natural history question to answer. Because it would be fascinating. I like all the studies that show the biases in our collections, and it would be really fascinating to have to find all of those duplicates and to see how they've differed based on their storage, based on a lot of different things. And I'm sure there are definitely well-documented cases where you might be able to do something interesting if they rigorously recorded where the samples came from.
Thanks.
Yeah, now that I think about it, and this is really, I may be totally making this up, and of course it's recorded, so you can check and make sure, and then you'll be like, Mason, you're making this up. But I think from that Buswell paper, 2011, 2010, the Australian Journal of Ecology paper, I think that they looked at, or they included duplicates in there and did an interesting weighting. Like, this is in their statistical model, basically, so they included all the data, but had it be such that that they did to avoid the pseudo-replication issue. But anyway, yeah, the thought just came to me that I've seen that before, but I've rarely seen that, the issue of duplicate, or how to handle or embrace, rather. It's not really a problem. It's actually a good problem, the duplicate thing.
Hey, Mason, one really kind of interesting thing that I thought was fun about your discussion about having an iNaturalist record associated with a herbarium record is another form of duplication, actually. Those will both eventually go to GDIF. And so you'll have literally a sort of a photo / specimen duplication, which I was thinking about it because we want to like integrate, herbarium data with iNaturalist data, and it's going to cause its own set of interesting problems as well as be like really amazing and important and valuable. So it's kind of fun to think about it from multiple lenses of duplication in 2024, right?
Yeah, for sure. And I've been on enough like iNaturalist kind of hikes where you see the whole group takes and uploads the same exact thing on iNaturalist. So that's probably actually pretty common too to have duplicates within iNaturalist too. So yeah.
So I had a couple more thoughts about the duplicates. So, and I think one of them gets back to something you said a few minutes ago, Mason, that I think we're not always sure what a duplicate is, because sometimes collectors will assign the same number to the things that are duplicates. And the way that I would interpret that is that they were duplicates made from the same original plant. But sometimes they will actually apply the same number to two different individuals. And so then you're left wondering, is this variation that's within a plant, or is this variation between plants from the same population? And so as you start trying to analyze trait data that you've taken from those duplicates, it gets tricky, I think, in terms of trying to figure out what it means. And I guess I'm wondering if anybody has any good ideas how we can use other information, and I know people have tried a few different approaches, but how we could use other information that's associated with either the collecting event or the other aspects of the collection record that we might be able to use to tease apart what sort of duplicate it is.
As an ichthyologist, it's always struck me as unusual that herbarium collectors don't image the plant while it's still alive as a routine.
Yeah.
We do that all the time to preserve natural coloration of fishes.
Yeah.
Yeah, so feel free to reframe if I want to go down Andy's Road. So this question, and you did have it in your list about, so maybe not only how it should be, data should be extracted, but the sort of, what does it say about our collecting practices going forward? What ought to we be collecting? This notion of, if we're going to use iNaturalist, taking the picture so we get the radial picture of a plant that's radial symmetrical before we smash it flat on the 3D sheet. So are there other practices that we could see in the future that would make the specimens even more valuable for research, and this kind of research is AI as well? So one of the things I saw that wasn't on the list that you made was the chewing patterns that I know people are doing work on, seeing who's been gnawing on the leaves, right? And so this notion of looking for those kinds of patterns and being able to see chew marks and bite marks and figure out associations and herbivory and other egg laying, things like that. So then just thinking, what does this mean in terms of changing our practices? Taking a picture, capturing close-ups, I don't know. DNA in the field, of course, people are taking leaf snippets. What else is changing, or what could be different? I don't know.
On that point, and related to why we don't image living plants all that often if we're going to be collecting a voucher anyway, I would want quantitative trait data from iNaturalist images, and that's not really possible right now. And even so, it's difficult with a 3D specimen, but then again, our herbarium images, they're distorted to an extent anyway when you press them. So I think it's fascinating that just computer science is kind of coming up with solutions to that. So there's a lot of really fascinating AI algorithms that sort of reconstruct 3D shapes from a 2D image. So I'm kind of waiting for that to be usable enough where you could get measurement. You could basically create a 3D version of an iNaturalist image again. So then you wouldn't need my little field prism template to get measurements in the field. You could just use the image. So I don't know if that's accurate enough, because if you're measuring a 2 centimeter long leaf, is it even accurate enough? Yeah, we'll find out someday.
Hi, this is Sharon from the Field Museum. To the question over here, what it made me think of was surrounding standards. So like the work that Darwin Core's doing with the event core and actually documenting the protocols, right? So that stuff's got to go with whatever standards are coming up with for this, or else it doesn't work. It's like it's not one standard on its own, right? And then what I was thinking about with what you were talking about, what can you learn from the geologists? The geologists do this all the time with a bunch of really big things that get squished flat, and they're reconstructing things in 3D, right? So I don't know whether there's a lesson that you can take from the paleobotanists who have to do this. They have even less than you do, right? A bunch of minerals that they have to sit with, picks and pull out. So that's what I was thinking might be useful there. But in the field, and I'm just looking over Doug's shoulder here at some stuff he's got on Morphosource, like the hand scanners now, people are, you can walk around with one of these things and get 3D scans of your living plant. So in terms of changing how you collect as a botanist, that might be something that comes along the line.
We also have a question online from an anonymous attendee. Techniques used to mount plant material on sheets varies greatly across herbaria and the individuals who do the mounting. What were some of the greatest challenges for machine learning caused by specific mounting practices? Which mounting practices could we implement that would help improve results from machine learning?
Oh, I passed it. You did pass it. Yeah,
And no, where was the overview? The big overview? The one slide I had. Sorry. I don't know. Maybe I didn't have it.
I don't think I know what you're talking about.
Okay.
Yeah. I thought you wanted to rule.
Oh, I didn't put it in this presentation.
Oh.
Okay, so one of the challenges that we wanted to address with Leaf Machine was using tape to mount specimens. If you just train a machine learning algorithm to identify plant tissue, then you end up getting a leaf that's bisected sometimes 8 times if they got happy with putting tape all over things. And there's definitely some collections that are more guilty of doing that than others, because like you say, different collections have different practices. With Leaf Machine, we kind of just had a blind hope that we could teach the machine learning algorithm to ignore the tape, and it ended up working. So I don't have a picture here, but like if you imagine that one of these leaves was bisected, if you go to leafmachine.org, you can see all of the, you can see an example of tape obstructed leaves and even stems. But we were able to teach the machine learning algorithm to ignore minor bifurcations around the leaves. I don't have a picture of it.
Yeah, okay.
Of tape.
Well, there's tape there.
Yeah, so something like this. If you were to imagine a piece of tape going across a leaf, other techniques were teaching the machine learning algorithm to identify just the green parts of the leaf, so to speak. But a lot of machine learning algorithms take the overall context into account now, so it's not purely texture-based. So when we created segmentation masks for our leaves, we just pretended the tape wasn't there, and it worked out. And now Leaf Machine just ignores the tape altogether, and you just get a nice little straight line. So it's not perfect, but it's a whole lot easier than trying to create a template matching approach or sort of trying to reconstruct that mask like after the fact, because you can't predict. Like sometimes it's one piece of tape, sometimes it's 10. It really depends. So if you can just ignore that up front, that's the best case. We did it just because I thought it would work. It turned out that it did. Sometimes you just have to try it and see if it'll work out. But yeah.
Yeah, I have an interesting, I guess, side discussion in regards to like, well, what is the best way to mount a specimen, I guess, for future use? And here I'm thinking, from my perspective of the non-computer, just like the physical material and sampling the physical material, say for leaf nitrogen or something, the ones that are linen strapped are like the easiest because you can actually just like, they're just free floating on there. So you can take a piece off for whatever you need. Whereas the ones that are glued down all tightly, that are great maybe for a lot of your work, are horrible for sampling because they're glued down and you don't even know if it's a water-soluble glue or what, so then it's just like, so anyway, I just had that aha moment of like, here I'm thinking, but then I know a lot of people too are also like, well, linen strapping, yeah, that's wonderful, but it also is like more prone because it's easier for me to sample. That also means it's more prone that over time for it to start to fall off, and the conservation of the specimen long term is not as good than as if it was potentially glued down. So yeah, so how do you mount a specimen then?
So there's another question online from Nikki Nicholson for Will. Do you envisage Flora Prism style images being uploaded through iNaturalist? Thinking about what can be captured in the field with existing systems.
I'm hoping that we can sort of scale that up in the future. We've been thinking about doing sort of like a Michigan census using that as a proof of concept. I'm working on like a really small project right now where I'm using it just to measure a lot of leaves really fast. So I'm hoping that scales up. That approach is nice because it is really simple. So if you go into the field, you literally just print out a piece of paper, take it with you. We print it on like shipping labels, so it's doesn't get damaged if it gets wet, things like that. But yeah, I'm hoping that catches on a little bit, at least until maybe some more 3D focused approaches catch on. That also kind of gets at a problem that we have, which is scalability versus precision a lot of the time. So you could go into the field with a 3D scanner and scan everything, but that scanner costs money. You have to train someone how to use it, and now you have a new collection type, which you're going to get great data for. But at the same time, we already have 1 billion leaves sitting there that don't have that. So it's, again, I'm not saying don't do it. I was saying we have to do it all. And it's a matter of what's best for the data that you have. Yeah, so I don't know. And then with respect to mounting leaves, that is a strange thing. It kind of depends. There are some mounting practices you can get around. So like we were able to overcome these little tape strips, but I am going to do it. Basically.
Oh, yeah.
The ruler problem. Okay, so this is something that's a little harder to overcome. So if you want to get measurements from an herbarium specimen, you need to figure out what a pixel translates to in the image. So these are just some of the rulers we encountered when we were making Leaf Machine. The worst one I've ever seen is this one, because it's a 1/4 centimeter. Whoever thought it was good to use 1/4 of a centimeter for a base 10 measurement, I don't know. But anyway, so for Leaf Machine, we had to come up with an algorithm that can basically deal with all of these. And the problem is you get something like this, where you have some rulers that are very close. So the 0 line is basically what a human said, using like an ImageJ style, just putting points on tick marks. If you use some of the rulers in the previous image, you get really high deviations from automated approaches. So these 2 rulers are my favorite. They're very simple, they're straightforward, and you can cross-validate measurements so you know you're getting the right conversion factor. So you always know that there's 2 points here, so you have a half centimeter, you have a full centimeter, you have millimeters, you can cross-validate. So this is something that's a little harder to overcome on the end, on the other side. So some of these rulers, you have to make an algorithm specifically for it, which is not fun. It's possible. But that's something you have to think about with all these collections. If you're making something bespoke, how machine readable is that? How translatable is that to someone else's data set? Are you making more work for someone 10 years from now? And again, it's hard to figure out if you are, because like we were able to overcome leaf tape, but something like this is a bigger challenge. Yeah, and putting 2 rulers in an image is also not really a fix, because if you get 2 different conversion factors, which one's right now? So you're kind of adding more problems sometimes. So yeah, that's my little soapbox rant. But if you need to put a ruler in your image, just do one of these and call it a day. But anyway.
Okay, so I had another comment about the different mounting approaches. So the fact that sometimes using glue versus some other medium, maybe you think, okay, maybe for measuring this or that, it's not going to make a difference or something like that. But for some of the types of traits, that, particularly the ones you mentioned early on, Mason, it seems based on some preliminary data that it can really matter whether there's glue on the back or not. And so Janine Cavender-Bears has been doing these really nice explorative analyses of spectra. And it totally depends on whether it's a fresh leaf or whether it's a leaf that's mounted, or if it's mounted with glue versus tape, and all of these sorts of things. Maybe algorithmically you can sort out some of that background later on, but at least early on you have to recognize these as sources of variation that you can then start to investigate. And, who knows? It might end up being too messy to be able to use our legacy collections, and it might just be something for collections of the future. So I don't know, Mason, if you've tried some of those, tried doing some of that too. It's just curious.
I haven't, the spectral things, but I've been like watching it, and yeah, a lot of the papers are really cool. But I think now that you mention that, like a lot of these tests are kind of like herbarium-like material, like it's dried, but it's not mounted. So now adding in that other layer of like, well, yeah, I didn't even think about the glue aspect until you brought that up, and I haven't done that work, so I haven't even thought about it. But yeah.
So Nikki has put up a duplicate set in the Q&A that she found on GBIF that shows a bunch of different mounting styles. I don't know if you can bring it up.
Sure.
Click on the Q&A down at the bottom.
I might need you to.
Down.
Oh, we don't see.
Oh, you don't? There we go.
No, I probably missed it.
There it is. Are you driving? Can you click that link for us?
Sorry, what? Oh, yeah, that's not on my screen now. It's weird.
Oh, but we can click it.
I have to click it over there. Okay. Is it over there? What do you mean? You do it then.
Yeah, there you go. See, you did it.
I did.
Ta-da!
I meant to do that.
Maybe you control plus there.
Yeah,
Maybe you
I
can
don't know
actually.
what's going on.
Somebody clicked on. Somebody's doing it.
Okay, that's not me. So,
This is showing the variation in.
Oh yeah.
Nikki's pointing out. Some of these are glued, some are taped. If you go back, the back arrow.
It's controlling. Well,
I don't know who's driving.
Oh, other people can't see what we're seeing, though.
Yeah. Yeah.
Folks online, you should be able to get get to this duplicate set by clicking on the link in the Q&A.
Yeah. Okay, it's in the Q&A. Yeah, all of the different various mounting techniques. Yeah, and I'm even thinking like, yeah, the overlapping leaves are kind of a pain for you, But for sampling, the overlapping leaves are great because they're usually not mounted. The part, so you can kind of just like take a little piece of leaf that isn't mounted. Cool.
I have a question related, I think. All of this to analyze the specimens on the sheet, there's also label data on the sheet. And I didn't know if any of this work, are you just concentrating on let's remove all the rectangles and the labels go away, or are you also trying to capture that data? Because I wondered if there were things we're learning about capturing label data, what we want to see on that label. Has any of that changed? Could there be any advantages to further standardizing of it? We know some herbarium labels are very rich with data, and then there are still data the ecologists, like the ones you brought up, Sharon, where they'd like to know more about things like methods, sampling protocols, things like that. How was the thing sampled? So are you removing the label data or are you using it?
Can I jump in, Deb? Is that okay?
Why not?
I know Will has an answer.
I don't know.
I'm sure Will has an answer for this as well. I'm really curious to hear Will's perspective, but I'll just quickly note that there's a tremendous amount of trait data, especially habitat. There's also, of course, associated taxa, right? And all of that can be assembled off of labels. It's non-trivial in a lot of ways, and we've experimented with trying to get kind of standardized data off of labels where we're really focused on those trait data and associated taxa and habitat. And it's a lot of it is just like the challenge we've been talking about for the last hour. Like you are trying to get those data assembled in a smart way off the labels, and then you have to standardize it, right? And you have to figure out how to standardize often challenging data. And I think all those things make it like a work in progress. But I do think there's remarkable data resources to assemble for labels themselves that we can get. It's just going to take a little bit of effort to figure out how to get it in a structured way that we can reuse and repurpose for the community more broadly. And I think that's just a big one to tackle. And so we're getting closer and closer to automating the steps for doing all that work, but there's more work to be done.
So more to Pam's question earlier, Brad Ruffel online says, at Michigan, when mounting, we put a leaf in a packet for future sampling if there is enough material available. This ensures at least one leaf is not glued down.
That's great. I love it.
Pam missed that, so I'll say it again. Yeah. Pam, Brad Ruffel says, at Michigan, when mounting, we put a leaf in a packet for future sampling if there's enough material available. This ensures that at least one leaf is not glued down.
Yeah.
Another comment from Nikki. She says, thinking about the digital extended specimen, would you assert traits onto specimens from literature-sourced taxon descriptions which cite the specimen, e.g. from monographs?
Like always, like, what does Will think? Yeah, I mean, that's a great thought. I'm guessing Rob probably certainly has a comment on that about, extracting trait information from, floristic treatments and species descriptions. I mean, I think I probably wouldn't ascribe it to an individual specimen unless you're certain it was measured from that individual specimen. But I think that, correct me if I'm wrong, more than I on this, more than me on this, Nikki, but like having it somehow though connected in this digital extended specimen framework, though, having I think most species descriptions will have like a mean and standard error of some sort, and having that at the correct taxonomic level. So I guess at the species level, not at the individual specimen level, unless you really do know that it was measured from that specimen. I don't know. Yeah, does that make sense?
I would say, this is Deb. Certainly in Taxon Works, we do that. So in other words, if in the literature it's cited, we make it possible for you to say this assertion that this leaf has this length or this whatever comes from this paper on this page, and it's associated with a species level understanding, right, of the species. If a specific specimen is referred to, materials examined, these are the exact things we looked at, then we also make that association with the physical specimen. But you can get both kinds of data, right, together. You can say, and it's asserted this is a literature-based citation versus a specimen's measurement.
And I guess that gets at the whole extended specimen idea, in that, a lot of us are doing work in our own physical databases to augment records with extended specimen information, but it's all manual. It's all hundreds and hundreds of hours of our work as collection managers to go and make those connections. Hopefully the extended specimen sort of infrastructure is going to make it possible to make those connections automatically without us having to be able to go in and do them in our databases ourselves. Which then brings up the question of, it's one thing capturing all of this trait data, but then how do you store it in a database, and how do you publish it out so that people can actually use that information? There's 3 separate steps there.
Yeah, and that gets to something that keeps me awake at night, which is when we start extracting all these quantitative traits, where does the data live? So I could have a database, it could go on Zenodo, it could go somewhere else in a less accessible format. It can link back, so like the Morpho source, you can link it back, but your server has to be there still. So I think all of us live in sort of a data fragile world right now. We definitely take a lot of these for granted, and they might not be here tomorrow. So how do we handle that? Because again, we all like something that is, an alive record, but it still might be nice to think sort of like the Internet Archive, and every now and then just stash a ton of it away, because you don't know what's going to happen. But yeah, so I think it is an interesting discussion, which is like, how do we want to manage all of this? So in some sense, it's great having something like GBIF. It's centralized, it's easy to use, but at the same time, it's difficult to add all of our traits back into it, and they don't want that necessarily either. So we have to figure out how to manage a fragmented world, but we still like having a GBIF API or something. So it's a really important discussion that I think Tadwig should definitely have, for sure.
Critical infrastructure that needs to be supported, for sure.
Hey, well, I guess I'd push back that GBIF doesn't want it. I just don't think GBIF can mandate like much of the structure of those data in a way that will make it interoperable and most discoverable. I think that this is a Tadwig conversation for sure. But there's I think GBIF has vast interest in helping to support the extended specimen concept and providing annotations to primary records. So those annotations can be multifaceted, including trait. But I think standardizing traits as a start is critical for allowing discoverability and interoperability. And we really want to be producing fair data out of the box.
Oh, yes, please, Rob. That sounds very exciting. I would like to say, yes, please bring the conversation, putting on my hat as past chair of Tadwig, the Biodiversity Information Standards, and reflecting back on a point that Andy added, is this notion of our respective databases. Whether they're homegrown, I don't know what they are, what you use, this notion of we have data that could certainly extend the knowledge of what we know about this thing. Whether or not we can get it out of our database in a structured way that makes it useful for others is still something where I think we're all evolving in that space. The number of so-called extensions to Darwin Core that people can use to share richer data, trait data, biological associations, formerly known as identifiers, genetic genomic data. It's one thing to say the standard sits there and says, here, if you'll just map and put your data in our bucket, we can do it. But the people don't always have the resources they need, either their database, or they need help with the software, or they need some sort of transformational help to get their data from one format into another. And that's certainly something we're interested in and are working on in TaxonWorks, is trying to ensure that you can get richer data out the door. But yeah, we're evolving.
There's a comment from Teresa Mayfield-Meyer online. She says, what database should it be stored in? And I think the ideal is with the extended specimen that it should be stored at the source. Integration rather than duplication. So we shouldn't have to duplicate all of that stuff. We should just be able to link it from the source and be able to link everything together.
Yeah, I guess I don't fully understand though, like what's the source? The home institution? Is that what you're saying? Or yeah, okay. Ah, gotcha, yeah.
I think that's really tricky though. I know the session's almost over. I'll just say like sometimes harmonizing and standardizing data is easier at an aggregator level, and then there's like round tripping between the aggregator and the sources to help improve across the board. I don't think it's a single answer there, actually, Andy. I think it's going to be like a lot of movement and like aggregators and more centralized data repositories will have a role to play, and the sources will have roles to play both, actually.
It's Sharon here. I guess to speak to that point, Rob, and something you were saying, where there is a push and a pull, and I don't want to be the cat and the pigeons here, but institutions look after collections, right? And we are aware that institutions then put rules around those collections. You have to make appointments. There are things you can't just walk in and take a specimen out. You have to follow the rules, right? I'm a database manager. I sit in an IT department. Ever tried to tell a researcher that they can't do whatever they want with their database? Now, if we are talking about doing this, then there are gives and takes around the database and the system that you put your stuff into. It cannot change every 5 minutes because you've come up with something new. And so there's a feedback loop between the researcher, the aggregator, and the institutional database. And institutional databases have to stay stable, right? Your research database doesn't. They're not the same thing. But I think often we're like, well, the researchers can move real quickly, so let's just put it in this new thing over here. And then now there are 50 things and we need to standardize them. They all need to have to come back together because nobody wants to use the institutional repository, which isn't quite so cool and sexy, but it holds data and it looks after it. And it's easier to share from that. The aggregators can pick it up. They can now say, we want more. And we've got to get an ecosystem where respect is given to all of those pieces. Just want to put that out there because if we don't do that, it makes it hard to share.
So I think coming back to this, the where to put it, what's the source? I mean, I tend to agree with the perspective of the source being the resource that's specialized for the mode of data that it's holding. And so GenBank, so sequences linked to the museum database from GenBank, morphology imagery from Morphosaurus as an example, or vocalizations from the Macaulay Sound Library, something like that. And I think there's because a single museum database or even an aggregator can't expect to sensibly pull in all the relevant metadata from such a diverse realm of data sets. I mean, maybe it can, but I mean, I think this notion of this extended specimen network is sure a network of institutions, but like a network of resources, differently specialized resources that specialize in either a modality of data or a level of data. So the museums, their role is, essentially to be the authors of the digital metadata in terms of provenance of the specimen. Now I think you can do something sort of in between where maybe the museums could ultimately physically or digitally host on their servers all the binaries, but then essentially have the metadata then network backed out to different specialized resources that can essentially properly or best provide access and discoverability and readability to all of that stuff.
Along those lines, Teresa online says, should a CMS have to store everything? I think not. Dan Glusenkamp also says, any prospects of all-purpose AI-driven database ingestion? Potentially we can have systems that can ingest your database, get some explanation from you in chat, and make sense of how to align your quirky system with a much more complex data lake. Should we invest in this rather than countless individual problematic database merging efforts?
No, this is very
Hi, Joe Mohan, UCI, or UC Irvine, but this seems like a good place to ask. So I'm coming from diatom taxonomy, which is kind of a land that has no trait database at all. No one really has thought too much about how traits interact with their environment. So I guess the big question right now in my head in terms of diatom systematics and traits is, what is square one? Because I think like plant land has a lot of these quirky systems, but I'm coming from like, what is the bare minimum? So, and how do I avoid many of these pitfalls to later integrate? Or do I just start integrated? So.
I don't know how to answer that. Does anybody? Yeah,
I mean, it's a great question. It's a hard one to answer because it sort of starts with like what you want to do with your trait database, right? Like, is it simply to like create a something that supports a research endeavor, or is it more like community building around defining like even just the traits that you're trying to capture themselves? Like, I mean, if it's a community building exercise, then talk to the community about defining traits and get the trait definitions that you need and how they're assembled from the literature or from specimens, right? If it's more like a research guided project, then like clearly you're going to have a core defined set of traits that you need. And I think like the questions become like what level are you assembling these trait data at? Like, or do you want to get like individual level trait data from individual level objects? Because that's a core level you need, or are you going to focus a level up at the species level? There's a whole bunch of questions I think are just critical to like just ask before you start. Like, what's the purpose? What traits are critical for your community? Those are the key ones that I think are starting points.
Yeah, that's great. That's super helpful. I think we're coming from a place where we have like really well-defined, obsessively well-defined traits and morphologies and very specific ways of describing species, but no like if you ask a diatom person, like how many species have spines, right? Like nobody knows that answer. But if you ask tree people, like how many species have needles versus leaves, like those are very basic questions that are simple for some groups, and just, you know, we're really just emerging thinking in this space. So yeah, I don't know, what questions do we even think about? I'm here to copy off of all your homework, basically, is what I'm saying.
There's nothing more online as of yet. Oops, hold on. Quickly before you go, another one from Dan Glusenkamp. He says, FYI, grants for collection rescue, backlog processing, and other work to rescue samples, specimens, collections requires a California nexus, must be in of California. FOST money for good work. callive.net/grants.
Very quickly, going back to what you brought up, I think, Will, this notion of Teresa Mayfield saying things like, where does all the data live? And then this question of how do we make sure the data is around in the future? And we talked about that at Tadwig last year, and I don't know what other people are currently doing, but this notion of protecting the data in the long term. So it's on a database, it's shared on some far-flung server on the other side of the planet. We also copy ours off onto hard drives that are not connected to the outside world, and we also try to print like on hard media, like CDs. We try to print on something that if the big mag solar flares come along or whatever, that we still have data, that we don't lose it. And so I think that's a good question about where does this trait data go, so all this work we're doing doesn't get lost in the event of something like that happening. I think that's a part of that question. And Teresa, to get back at your point, we also try to be very thoughtful. If we're going to store, like, we're not going to store your entire, whatever, your genetic genomic work, but we're going to do our very best job to store the data we need to store for you that helps you relate the information in GenBank or wherever else you published it back to the records that it's appropriate to in the database, not in the notes field. So we do make decisions about what we're actually keeping locally.
Actually, I have a question, I think maybe for Rob, and I think I should know the answer to this. But like where are we with sort of standardized terms for traits, for qualitative traits, or specifying feature, presence, and absence? It seems like ontologies like Uberon are a step in that direction, but essentially human and machine readable descriptors that would be easily shareable.
Oh, I think there's so much to do in that space that has not been done yet. I think there's like a lot of innovation coming at us at 1,000,000 miles an hour through like RAG LLMs and other ways that we're going to be able to capture a lot more diversity of traits and be able to structure those and learn how to like help have machines and humans like co-structure trait knowledge more effectively. We've learned working on a project called Futurists, which is focused on vertebrate traits, is that like it's a slippery problem in that there are vertebrate trait ontologies and there is Uberon, but they don't always link up in ways that you expect in terms of like reporting how an archaeologist would measure a trait, or a paleontologist would measure a trait, or a neontologist think about external measurements or internal measurements, and kind of like linking it all together and making it really work is incredibly difficult work for people who are really good at thinking about like ontological puzzles. And so I'm really hoping that like the machine learning, semantic AI, and the statistical AI methodologies that are kind of separately developed are merging and becoming more rich so that we can kind of use machines to help guide us with structuring our knowledge a little more effectively. I think it's a huge area. I know other people have thought about this, and then Will, you may have your own thoughts, but I think the bridge between semantic approaches and semantic AI and statistical AI is probably the most exciting thing that I see on the horizon. So we'll solve a lot of our problems with like bringing all the data together and learning how to structure it more effectively.
So Nikki says online, where does this discussion go next? We've mentioned Tadwig Spinach, but submissions closed for that event unless you already have a slot. Deb says maybe we can find a more unconference space at Spinach Tadwig to continue this conversation, capture ideas, or share work. Teresa Mayfield also says, how do we sustain the databases that are good for data such as traits?
I'm not going to answer Teresa's question because I don't know the answer to it yet. But for your diatom question, I guess the bit of advice I would give you as an old lady now, try not to build it on your own. Like, find your other diatom mates and build something together. Even if it's not the perfect solution, and even if it doesn't use something that all of us use, at least it's something you all use together. And then it can become part of a network. So that would be my simplest bit of advice. Like, if you really are starting from absolutely nothing, like, don't hive away in your little office and build it on your own. Find your diatom mates and build it together.
Thank you. Yeah, we have a consortium of diatom taxonomists, and just building a way that they can all contribute to like a trait space is kind of the goal, because they're largely not thinking like trait ecology type questions. So thank you.
And actually, that makes me think, if you haven't thought of it already, have a core record of your database be the specimen or the sample. as opposed to a taxonomy or something like that.
So I was just going to say, come talk to us about TaxonWorks. See if it will fit or give you ideas anyway. I think we can do what you're. Okay.
Any comments on Teresa's question? How do we sustain the databases that are good for data such as traits? What comes to mind immediately for me is the Action Center that we've all been talking about. The Action Center for Biological Collections. I see that as the perfect vehicle for being able to sustain this critical infrastructure that we need in order to be able to bring about this realization of the extended specimen, of being able to link stuff together, supporting things like Morphosource, supporting things like trait databases, supporting those kinds of critical infrastructure pieces that we need in order to be able to keep all of those linkages going to be able to bring about the extended specimen.
So you mean hardware and people?
Yes.
Yeah, that's what I was going to add too. That's all, just software. No, no, I'm not going to say too much about it now. But yeah, there is a. If anyone's interested, there is a series of presentations and panel discussions specifically on this question of sustaining data and informatic services tomorrow in the morning.
Teresa asks, does anybody know how TRI is funded? They mentioned it earlier in their talk.
Yeah, I don't know. And again, we're recorded, so maybe I should be careful, but every time I've uploaded stuff on there, it seems like it's just a few people, like in one institution that seems to maintain that database. So, yeah, but I don't know.
I mean, it sounds like a lot of people don't know what TRI is, so maybe that's the problem.
Yeah, maybe so. Yeah, I don't know. Maybe Rob, do you know?
I think so. Yeah, TRI is a really powerful resource. It's focused on plant functional traits, which has a relatively narrow delimitation in the way that TRI kind of conceives of those traits. I mean, I think there's a lot of work to capture traits that are more floristic as opposed to like kind of more leaf economic spectrum traits. But like, I think that different governments, different organizations at the intergovernmental level just have different working agendas around sustaining infrastructure and projects. I think that the German system or the EU system might have more support for sustaining these efforts, but I think they're all grant supported at the end of the day. I don't think many projects, some of them are philanthropically supported. I don't think TRI is. I think TRI has had relatively strong support from either the EU side or the German federal side for a long time. I also think like one thing I'll mention is like sustainability is often viewed through the lens of like organizations that take action. Like, the Action Center is a great idea. I would make a separate pitch. Reduce the cost of your infrastructure. Make it streamlined, slim, and inexpensive to run. We've run infrastructure that is highly used by the community, but we've really tuned lowering the cost of maintaining the infrastructure. That's easier said than done for many infrastructures. Morphosource stores a lot of 3D image data. That's expensive. But if you can get away with it, make it as cheap as you possibly can. Sustainability revolves around having resources and reducing costs.
So Teresa says online, this is the part that makes me crazy. These nice things can fall away if a single person retires or an institution decide it isn't important for its work. And many rely on volunteers or institutions that don't know what they're sustaining. The most expensive part of any of these resources is people.
Yeah, I will add, I do know that Try does have herbarium extracted data in there. Some. It's a minority, but they do have it, so.
So I am cognizant of time. The buses are going to be arriving at 4 o'clock and we'll leave at 4:30. It is now 10 to 4. So just putting it out there.
Okay. Yeah, I think this was to run to 3:30 anyway, so, but this is great.
You've done well.
Yeah. Cool. Any final words, or we're good, I think. Cool. Well, thanks everybody. It's great. We'll have to keep it going.